Hi. In this video we're going to talk about Chapter 9 of the textbook, Forest Ecosystems by Perry et al. And we're also going to talk about general concepts in forest ecology that go beyond what we may have been talking about so far. And specifically, what we want to talk about is a way of thinking about forest ecology that's been really critical to conservation science. That is thinking about forest ecology in terms of three broad categories of terms that all overlap and intersect with each other. And so if we think about how a forest works in terms of forest composition, forest structure, and forest function, then this triad can really help us understand how ecosystems work, what's critical to conserve, or what's critical to think about when you're thinking about management impacts on ecosystems worldwide, no matter where we are. So when we're thinking about composition, this is a question of who is present in the forest. And so we can think about conservation of forest ecosystems in terms of conserving individual species. The Endangered Species Act, for example, focuses on this in the United States. We can think about ages. For example, this has been really important in forest conservation in the Pacific Northwest because we think of old growth versus young stands. So composition doesn't just have to do with who's there, but what age individuals are present. So we're talking about demographics here. Similarly, for plants that differ in sex, you could think about what, whether you have males or females in plants on the landscape. Finally, composition is going to address how many of each. What is the relative abundance of any given species in an ecosystem? How equally is space shared? It's not just how many do you have, but are all of the resources and spaces equally shared within a given ecosystem? So that's all composition. There are a number of ways in which you can think about composition, and we'll go through a couple of those. One of the more common ways that we think about composition is in diversity indices or in ordination plots of composition. Then we have structure, and structure is probably what you've mostly been focusing on so far in any typical forest ecology training. And so our structure is often going to focus on things like structural diagrams like this, right? Excuse the blur there. Where we're going to focus on horizontal structure this way, but we might also focus on vertical structure in the canopy. And those are often represented by vertical structure diagrams, and I'll give you a couple examples from your book. We also have above versus below ground, so we can think about those types of structure. Here's an example of these vertical structure diagrams that are common in forest ecology texts. In this case, this is showing leaf area as you ascend with height into the canopy. And then, of course, we have structure that integrates horizontal structure in multiple dimensions. And so you can think of your maps or kind of GIS applications showing heterogeneity of forest density, for example, as being an example of structure. So structure, we can go up and down, we can go side to side, and then of course there's above and below ground elements. So how much material is above ground, how much material is below ground in an ecosystem. Those are all important components. And then finally, let's talk about function. And so some of the oldest forest conservation movements in the United States as a nation go back to the desire to preserve ecosystem function in terms of watershed function, watershed storage. And so hydrologic function is fundamentally important. These days there's a lot of discussion about carbon storage in forest ecosystems. It's been central to global approaches such as the IPCC reports with regard to changing global climates. So ecosystem carbon storage is a very important function. Productivity is related to that, but a more traditional way of interpreting ecosystem function in terms of the ecosystem bringing carbon and energy into the system. And then we have nutrient cycling. One of the first things we think about when we think about forest composition is we think about the diversity of a given forest. Now, of course, the diversity of a forest is first predicated on how many items there are, right? So, or how many species there are. So we first think about species diversity. How many are there, right? How many? This, of course, is species richness. Okay, so species richness. Now, I've drawn two cross sections of a forest here, and clearly they are different. However, if we just count the number of species, these two forest cross sections would be equal. Obviously, these are not equal, and so in order to deal with diversity, we have to do more than just count the number. This is the idea behind species evenness. So the concept of species evenness, clean up my E here, let's capitalize that, it's very important. The idea behind species evenness is that we're saying how equal is space shared. So here I have two species and they are sharing the space perfectly, so we have an evenness value of 1. In the case where I just had all red species and only one green species, then our space is very unequally shared and we have low species evenness, even though we still have a species richness of 2. If we want to combine richness and evenness, more commonly we will use an index of diversity, something like Simpson's, or more commonly Shannons H. So Shannons H is an index of diversity that combines values of richness and evenness into one value. And here's how this typically works. We generally take the negative sum of the proportion of individuals in a group, in an area, in a forest, that are of a given species, times the natural log of that same proportional abundance. And so by doing that, if we have more individuals, more individual species, then we have more numbers like this to add up. So we do this once for each species, and then we add them all together. So if you have 50 species, then you have 50 values like this to compute and add together. Similarly, if these proportions are higher, so if each individual makes up each species is made up of individuals that make up a large proportion of the total number of individuals of all species, so the proportional abundance is higher, then each of these values is higher, and your Shannons goes up. So we get a higher number of Shannons as we combine those. So that's our Shannons H. The higher it is, the higher diversity we have. If that negative confuses you, consider that the natural log of 0.5 is around negative 0.69 or negative 0.7, and the natural log of 0.2 is about negative 1.6, so these are larger negative numbers. We talked before about other components of species diversity, which might include age, demographics, right? These are important components of diversity. One thing we haven't talked about that's increasingly also very, very important, critical in fact, especially to northern temperate ecosystems where you have very few species, is we can talk about the genetic composition. So not just what species do you have, but what genotypes do you have? What are their genetics? What's their selective history? And what are their adaptations to a given spot? So we can't just stop at the species level, we have to go beyond that. We could also go the other direction, and instead of thinking of genetic composition, we could think about genus level. What genera do you have present? What plant families, for example? If we're just thinking about plants, we can think about families. So when we're thinking about composition, we have to go beyond just thinking the species level. We have to think about all of these things. And then finally, of course, we have trophic levels that we have to think about. Okay, all the way from the predator to the primary producer to the decomposer, all these elements of composition are going to be fundamentally important. When we think about composition on the landscape level, we have to go beyond just thinking about how many or how diverse the total assemblage is, and we have to think about how it changes depending on the landscape. A fundamental concept in ecology is the idea of a species area curve. And the idea behind a species area curve is that as you increase area surveyed or sampled, you also increase the number of species. At some point, for a given ecosystem type, you level out and you end up with kind of a maximum number of species for that given system. When we first start sampling, there's a minimum sampling area you might decide on for a minimum location that you can look at, and that is the location that, say we start sampling here, a single sample might have a value here, and we would call that our alpha diversity. So alpha diversity, as at an individual scale, the smallest area that we would sample, how many species are present. A given, it might be a stand, it might be a given sample plot, depending on the context, right? But alpha diversity is in a given small location. Gamma diversity, on the other hand, is what happens when we sum up all of the species within a given area. So all of the species summed up, and we end up with our gamma diversity. How this curve changes through time, we refer to as our beta diversity. Or not through time, through space, I'm sorry, as we expand space. How this curve changes, we call our beta diversity. Another way of thinking of this is the turnover as you go from one plot to the next plot. How many additional new species do you get, and what's the shape of that curve? Let's give a better example, because right now we're looking at one curve when in fact we could be looking at many. A typical ecosystem might look something like this, where we're accumulating species relatively rapidly. Our species richness is going up as we sample more area, and then we're leveling off through time. But another possibility would be an ecosystem that would do this. Instead of accumulating species rapidly, species accumulation is relatively similar as you expand area. And so you're accumulating the same number of species, or similar number of species as you go through time, and then again eventually reaching an asymptote. Similarly, you could have another ecosystem that more rapidly accumulates species and then levels off. The shape of these curves can be predicted somewhat by ecosystem heterogeneity. And so it interplays with the geologic history, the landform history, the geomorphology of the area, and the dominant species that are structuring the system. So a foundation species, an individual species that is having a big impact on the structure of the system, can influence the heterogeneity, canopy gaps, open spaces, microhabitat diversification. And if you have a lot of that, then as you move from place to place, each place is very different, and you'll accumulate new and novel species before leveling out. On the other hand, if you have a very homogeneous ecosystem, as you move across the system, you're going to slowly pick up new species depending on dispersal or more demographic trends, less than microhabitat heterogeneity, and then most species are somewhere in between. Okay, so here we have it. This is alpha, beta, and gamma diversity. When we think about who's on the landscape, we might also divide it into thinking about the roles, not just their roles with regard to trophic position, but we might think about the roles with regard to something called keystone or foundation species roles. These two terms are not always talked about as mutually exclusive. A keystone species is probably something you're familiar with. The idea is that it's a species, often predators are invoked in this role, that plays a fundamental role in holding the ecosystem together that is disproportionate to their biomass. Foundation species, on the other hand, are often proportionate to their biomass. These provide structure for the ecosystem. Typical example of a keystone species from northwestern forests would be something like perhaps a spotted owl, predator species such as bears, wolves. These are often keystone species. They play foundational structural roles. Wolves in particular have been implicated in this in riparian forests of the West, where they keep herbivores away from riparian areas where herbivores otherwise will heavily browse willow and cottonwood species, resulting in fewer trees near stream sides, and they alter hydrology and stream function and fish habitat. Whereas a foundation species you can think of as something like a Douglas fir, which provides a dominant position in many forest ecosystems, structuring the canopy and structuring the whole ecosystem. So foundation species versus keystone species. Structure is most frequently thought of in terms of both cross-sectional or horizontal structure and vertical structure or canopy structure. Canopy structure has been emphasized a lot in recent years because it was a proportion or an area of forest structure that was long neglected in forest ecology due to limited ability to access the canopy. But now, due to a combination of satellite detections, of course, air balloons that allow people to access the canopy by floating into them. That's my drawing of an air balloon. Excellent, I know. Canopy cranes, which allow people to ascend into the canopy in buckets where they're carried into the canopy. And then, of course, climbing gear. Okay, excellent drawings, I know. This has allowed us to do a much better job now of sampling canopies, and so in many ecosystems we have a better idea of vertical structure. The graph I've shown here might be two species, where the red bars represent one species, the green is another, and the amount of leaf area for each species as you go up in the canopy is represented by the length of these bars horizontally. So you can see that we have one species that occupies a low canopy position with the majority of its leaf area, and then we have another species that represents the upper part of the canopy. Okay, so that's our vertical structure. Very important for thinking about especially old growth forests, also important for thinking about fire behavior and interactions with disturbances. Horizontal structure is more traditionally considered by forest ecologists, and these sorts of diagrams have been in the literature for over, for hundreds of years, obviously. So this structure is depicting the average horizontal arrangement of species and size classes within an ecosystem. So, very important. But another component, another kind of frontier of thinking about structure in ecosystems is the structure below ground. It's often thought that below ground represents about 20% of total biomass. Another way to think about it is it's approximately 25% of the above ground biomass, if you know what the above ground biomass is. This is a gross assumption that is based on how difficult it is to see below ground and determine the actual amount of root area for any given plant. What we know is that some plants actually have very different, and dominant trees especially, have very different rooting behaviors, very different rooting strategies, and that structure can have a large impact on who's below ground. For example, taking up water resources that aren't available to seedlings of other species will affect the composition. If a tree is a wide, shallow rooting individual, then it's going to affect who can grow beneath it. How aggressive those roots are will determine the composition of other individuals within the forest. On the other hand, trees that are deeply rooted might be tap-rooted into more permanent or more reliable sources of water, which interacts with ecosystem function. So the below-ground function, or the below-ground structure, can have a big impact on ecosystem function. Similarly, the fine root structure has a lot to do with the ecosystem, both in terms of composition and structure. We can also think about structure of other organisms, including fungi, root-feeding organisms, other organisms that live below ground. So there's a lot more to go into here in terms of structure. On the landscape scale, it's fundamentally important to think in terms of structure, not in terms of just those vertical and horizontal images, but we also have to think about the arrangement of different types and patches of systems on the landscape. For example, a patch of one type of forest here, if we're looking from above down at a map, and a similar type of forest here, surrounded by a matrix of different types of ecosystems. I'm mixing up all my markers now. Might result in our green dots here, our green habitat patches here, and the squiggles I'm drawing here are going to represent different ecosystem types or habitat patches. Okay, we can think of those green dots as being essentially islands of habitat for certain organisms. The arrangement in terms of how close they are and how large each individual is very important for conservation. Similarly, the presence or absence of corridors of habitat connecting those two ecosystem types will be fundamental. And so we don't just think about ecosystems in terms of a single ecosystem and its composition, but we have to think about it in the context of other ecosystems and the landscape arrangement of patches of large and small size. A key way this has come into conservation design is the idea of core versus buffer areas. So for example, for old growth forest, there have been proposals to structure conservation areas such that you have core, untouched areas surrounded by buffer areas where you have intermediate management, and core areas should be connected by a corridor. This is important for wildlife. It's also important for plant dispersal. One place where landscape level conservation, buffer, core, habitat heterogeneity issues have all come to a head is in the debate over conservation reserve design and forest ecology, called the SLOS debate. The idea behind this debate is it better to have SL, single large, or several small reserves in conservation design? So here's our example of several small patches that are all spread over the landscape, or single large patches that have minimal boundary area. And so there's a lot of debate, and it depends on the organism which is better in forest conservation design. Of course, one ideal design would be to have a lot of buffers and corridors between individual, single, small, several large sites. Okay, how many lines can I draw here? I don't think I'm done yet. Okay, single large versus several small. And finally, let's talk about ecosystem function. So ecosystem function, as we brought up before, might have to do with the amount of sea storage an ecosystem has. It might have to do with nutrient cycling. It might have to do with hydrologic function. Each of these, as are many of the topics that we're covering today, could be its own lecture, its own entire course. So there's a lot to talk about in each of these. So these are primary functions of forest ecosystems for sure. Another one, though, has to do with habitat provision. The structure of the ecosystem in terms of how it's arranged on the landscape and the composition of who is present all are going to determine the nature of the food web or the interaction between nodes in connection networks within an ecosystem. The nature of these interactions are fundamentally important for forest ecosystems, and they interact with other components of ecosystem function. Nutrient cycling and carbon cycling are fundamentally dependent on the nature of energy flow in this food web. And so the amount of primary producer material that is eaten by an herbivore is going to determine how much energy is transferred to that next trophic level, how quickly nutrients are cycled by and aided by eating and digestion of the herbivore, and how quickly that'll go on to other organisms, how much material is available to detritivores. This all interplays with the rest of what we're talking about for ecosystem function, so a lot to talk about here. Several excellent examples from your book cover food webs in all of their great complexity, and they can be incredibly complex. Increasingly, sophisticated methods are being used to dive into these sorts of food webs and measure things like the connectance among nodes. How many nodes are connected within an ecosystem can determine its ultimately determine its connectivity, which has a lot to do with how resistant that system is to disturbance and how resilient it is following disturbance. It's important to point out that one of the main reasons that we have such a tight connection between the nature of a food web and the structure of that food web and these ecosystem functions like carbon cycling and nutrient cycling has to do with energy transfer efficiency. So in general, it's thought that most organisms are only going to have about a 10% energy transfer efficiency when consuming the next level, the next trophic level. Below or above them, however you think about it. So, for example, an herbivore that eats a primary producer is only going to be able to assume about 10% of the biomass, only about 10% of the biomass it consumes will end up as biomass in the next level. So an herbivore has to eat 100 pounds of primary producer in order to produce 10 pounds of herbivore biomass. Similarly, a predator has to eat 100 pounds of herbivore in order to produce 10 pounds of predator biomass. However, the requirements for nutrients are going to differ among each of those levels, and the interplay between the stoichiometry of nutrient dependence combined with energy transfer inefficiency or efficiency, however you want to think about it, results in nutrients sometimes being available as you transfer from one level to the next. And so the herbivore may consume many more units of a given nutrient of a producer than it needs in order to produce a certain amount of biomass with some nutrient left over, depending on its stoichiometric balance, the balance between its carbon, nitrogen, and phosphorus principally, but other nutrients as well, right? So the interplay between all that has a big impact on carbon storage and nutrient cycling. And in a future lecture, we'll go into detail with an example there where we can trace the carbon transfer efficiency and the nutrient cycling between levels within a food web. Finally, let's talk about some fundamental concepts within communities. The guild concept is fundamental, and hopefully you've learned about that in earlier ecology classes. This is a classic example from MacArthur, 1958, where different warbler species were seen to feed on different locations within a tree canopy. The idea was that these spatial feeding patterns represented distinct niches, and species were able to have different niches within the exact same ecosystem, and those niches allowed for coexistence. And so this is fundamental to coexistence of species in ecosystems. Because guild members overlap in their functional role, of course, their competitive exclusion can occur. And so within a guild, if species are too similar in their niche, then they can outcompete each other. And this is a fundamental cornerstone of thinking about ecology, that no two species can perennially have the exact same niche without some diversification occurring. Finally, one closing concept that's important to focus on is the role of genetics and the maintenance of genetic diversity within populations. This is an easy thing to forget about when we're thinking about management on the landscape. Planting new individual trees on the landscape in a forest ecosystem is a genetic experiment. It is interface with the genetic diversity of previously established tree species on the landscape. And so it's important to think about what actually maintains genetic diversity within populations and why it's important. So for our first point, environmental variation favors different genotypes at different times and in different microenvironments. Similarly, individuals that are heterozygous are more fit in variable environments. So in addition to having an environment that can be variable, that selects for different individuals across it or through time as the environment changes, if the genetics of an individual species is quite variable, then that allows for some adaptive variation, or variation that can take advantage of distinct environments and distinct times. Heterozygosity in genes is generally thought to be something that improves individual fitness because it protects against inbreeding depression, which can be fundamentally important, especially for small population sizes. Within tree populations, we don't have to think about this often as much as we would for animal populations that often govern the management of forest ecosystems. So, for example, the inbreeding depression in spotted owl populations that are dwindling rapidly in old-growth forests might affect the management of forests that are populated by Douglas fir that is in no danger of inbreeding depression due to the high number of individuals on the landscape. Another factor that plays into maintaining genetic diversity within populations is what we call neutral mutations, genes that can be maintained in the population with no clear adaptive role. And part of this can be that genes are maintained within individual species as what we call ghosts of selection past. That is, they had an adaptive role in the past, and they may not have an obvious adaptive role now. Similarly, you can have genes in individuals that control traits that just simply have no clear adaptive significance, or they go along for the ride with other traits that have more impact. And so in order to have a given trait, maybe a given type of feather, a given color, another trait often comes along with that that is of no particular adaptive significance. Genes may be at a selective disadvantage in some environments, but they can be maintained in the population, especially through interbreeding with populations in other environments. So wind pollination is a classic example of this for forest trees, where we have forest trees that may have been selected as saplings or as young seedlings for one particular microenvironment, but as they grow up, in order to interbreed, if they're dependent on wind pollination, they may receive pollen from trees that are in completely different populations. Again, pine trees, most of our conifers would fall into this category, and because of that, their offspring may have traits that reflect both the selective environment of the maternal tree, of the mother tree that has the cone that drops the seed, but also might have traits of the paternal pollen that came from a completely different environment. So this is particularly common in mountainous environments, where you can't expect that just because a tree is producing seeds at a given location, that those seeds will be adapted to that location. It depends on both the maternal microenvironment, selection pressures, and who contributed the pollen.